Hello students, welcome back. So we have started with the new module, that is GI physiology, gastrointestinal module, and this lecture is just a warm up for you guys to just wake up and know general principles of the gastrointestinal. So the learning objectives are, by the end of this lecture you must know what is physiological anatomy of GI wall and enlist functions of the gastrointestinal smooth muscle, and then describe certain physiological characteristics of slow waves and spike potential. So digestive tract, also called as alimentary tract, provides the body with continual supply of water, electrolytes, vitamins, nutrients, etc. So how does it do that? If we say that what would be the functions of this digestive tract being brought, you have a tract on its own, and then you have accessory organs that aids this tract, alimentary tract, in its function. So digestive tract is long tube, you can say, from mouth to anus, and then you have these accessory organs which includes teeth, tongue, salivary glands, liver, gallbladder, pancreas, which all aid in the functions of the alimentary tract. So what would be the functions of the alimentary tract? Of course, movement of food through it, and then the secretion of digestive juices and digestion, you can say digestion of food, and then you have the absorption, because all of the things are absorbed from your alimentary tract. So absorption of different important electrolytes and digestive products. And then what would happen after absorption? Of course, circulation of it through of these absorbed substances in the blood, and then control, of course, of all these functions is through the your nervous system, which is through the enteric nervous system, which we'll be discussing in the next lecture. So you can see this is your digestive, you can say, tube or tract or alimentary tract, starting from your tongue to the ending up in the anus, and it is approximately 30 feet in a cadaver. So all of you know the basic structures of the digestive, the alimentary tract, and then the aiding organs, for example, the liver, the pancreas, and your salivary glands, the oral cavity, and all these structures. Everybody's knowing that. So this is an important diagram in this lecture. For, of course, you are just showing the walls of the GI tract, and if you come from, if you go from outer towards the inward, so you have, then you have serosa, and then you have muscular layer, which is longitudinal muscular layer, which is on the outside, and the inner you have the circular muscular layer, and interspersed in between these layers you have your Meissner nerve plexus and mesenteric nerve plexus, which we are not going to touch in this lecture. When we're doing the enteric nervous system, we will just look into it, what's this. And then you have the mucosa, the mucosal lining, and the submucosa. So if you start from inward outwards, similarly you have the submucosa, you have the mucosa, and then you have the circular muscular layer, and then you have the longitudinal muscular layer, and the outer layer, which is the serosa. So here, now given in the order, serosa, which is also called as peritoneum, and then you have longitudinal muscle layer, which was outside. So you call it externa, and then the inner circular layer, that's why you call it as muscularis interna. And then you have the submucosa, and then you have mucosa, and mucosa still further have three subdivisions, which is muscularis mucosa, lamina propria, and the proper epithelium. So coming to the characteristics of the mucosa, this is the innermost layer of the tract, and it is composed of the layer of epithelium, which is usually simple columnar in most of the GI tract. However, it is subject to change in different areas. Then you have the lamina propria. This is a loose connective tissue containing blood and lymphatic vessels, and then you have the proper muscularis mucosa, a thin layer of smooth muscles, uh, uh, which is responsible for different muscular folds or rugae that serves to increase the surface area for digestion. And this is the most highly differentiated layer of the GI tract because of tissue specialization, that is differentiation across the GI tract. Submucosa, It is loosely bind to the mucosa to underlying muscle layer. It contains of different blood vessels, lymphatics, nerves, and plexuses. And then you have muscularis. I told you we have external and internal layer, and this is the thickest of all the layer. It begins at the mouth, where it is composed of mixture of smooth and striated muscles, and external sphincter, where it is skeletal. So the distal pharynx, it turns into all smooth muscle that courses throughout the rest of the tract. So when we will be doing each part separately, right, starting from the pharynx up till the anus, we will be discussing each part separately. Then we have a smooth muscle layer, and the smooth muscle is very important because all of the motor functions of the stomach of this everywhere, but the GI tract are usually brought about by the smooth muscle layer. It consists of an inner circular and outer longitudinal layer. And then we had the serosa, which is the outermost layer. It is composed mostly of a thin layer of areolar tissue topped by serous membrane, mesothelium. It begins in the lower 3 to 4 centimeter of esophagus and ends up in the sigmoid. So just go through the text here and you can see the different layers, the inner circular layer and the outer longitudinal layer, and you can see the its orientation. Then you have two plexuses, that is the orbars and the Meissner plexus, or the myenteric and the submucosal plexus. These are the nervous connections interlaid within these layers. The parasympathetic vagal fibers, we were discussing this in the enteric nervous system. So coming to the GI smooth muscle cells, which you must know them properly because much of the motor functions of the gut, they are performed by the different layers of the smooth muscles. Now the smooth muscle, usually they function as a syncytium, that is, if one fiber is activated, all of them are activated because they are joined together. So the length of these smooth muscle fibers, their diameters, and they are arranged in parallel bundles. And then we call them longitudinal muscle layer because they are spread about longitudinally, and we call them circular muscle layer because they usually spread around. Now, the GIT smooth muscles are excited by continual slow intrinsic electrical activity along the membranes of the muscle fibers. Now, this diagram is very important. From this diagram, you can do all of this. I guess this is a summary of this chapter as well. And this is one of the most important thing, that is the slow waves. There are two types of waves. Waves, what do we say wave? Because these are undulating. These are not action potential. So we have two types of basic type of electrical waves in the GI smooth muscle. One is slow wave, one we call as slow wave, and one is called as spike potential. You can see the spike, these are action potential. These are actual action potential. And the slow waves, these are undulating waves. They just go just above the resting membrane potential, and then they just come back down. So you must know the physiological aspects of uh, of both of them. So most gastrointestinal contractions occur rhythmically, and this rhythm is determined by mainly by the frequency of the so-called slow waves. For example, the slow waves would be different in different aspects of GI tract. For example, 3 in the body of the stomach, then 12 in the duodenum, or 8 or 9 in the terminal ileum. So they are different. Now, the precise cause of the slow wave is not completely understood, but we can say this is because of different type of smooth muscle cells that are specialized cells, which are called as the interstitial cells of Kajal. Okay, Kajal. This is not Kajol, that film actress. This is Kajal. These are in this interstitial cells of Kajal that are actually muscle cells that are interspersed between all of these. You can say the smooth muscles, and these are the electrical pacemakers, guys, for the smooth muscle cells. So these interstitial cells form a network with one another and with other fibers, smooth muscle fibers, and they have synaptic connections. And what happens if these cells undergo cyclic changes, these cells of Kajal? So the membrane potential, due to their unique ion channels, they periodically open and produce inward pacemaker-like currents that may generate slow wave activity. So the slow wave activities are formed in this way. So the slow waves usually they do not themselves cause any muscle contraction in most of the I guess GI tract, except perhaps in the stomach where they might cause. Instead, they mainly just excite appearance of intermittent spike potential. So they would just excite the formation of the actual action potential where they may cause contraction by themselves, they may not. And these slow waves, guys, you can see the resting membrane potential in GI usually minus 56, but can range between minus 60 to minus 50. And minus 40, you know, it is a threshold. And once it goes above minus 40, so then you are going to end up with a spike potential. And you can see these slow waves, they just go above, not even above minus 40, somewhere between minus 50 to minus 40. So these are undulating changes in the action potentials, in the membrane potential, sorry, which causes the formation of slow waves. And I told you this is because of the special characteristic of the cells of Kaju, which are present within the smooth muscle. Then you have the spike potential, guys. The spike potential, these are action potentials. Action potentials, you all know, that would be caused because of the depolarization. Now, one thing is different to guys over here. The depolarization is not because of the sodium channels, fast sodium channels or voltage gated sodium channels. Rather, it is because of the calcium sodium channels. That is, a lot of calcium would enter and some sodium that causes these channels to open late but close slowly. So if we say what's the difference between the spike potential, action potential here, and the nerve muscle, would be that in the nerve, the action potential would occur abruptly and finish abruptly, whereas the action potential in GI, in spike potential, it would be occurring late, but then it would be sustained contraction because of the sodium, calcium sodium channels. So slow waves, they cause rhythmic gastrointestinal contraction, and these are not action, as you all know, and this is because of the special cells of Kajal. They have unique ion channels that produce these. Now, spike potential, I told you these are the action potential, these are true, and these spike potential are generated each time when the peak of the slow wave becomes more positive than if it goes above minus 40. So what's the difference between GI smooth muscle and nerve fiber? I told you nerve fiber action potential are caused by sodium ions through, whereas here we have calcium sodium channels. These are the new channels, and the movement of the calcium ions through the interior of the muscle fiber during action potential causes them to contract. So calcium ions, then you all know what calcium ion does, the calcium calmodulin complex, and then there is a activation of the actin and myosin. The active sites are exposed, and then you know that myosin heads, they have ATPase activity, and then there is an attraction between them and the contraction occurs. So if we, this is important, how do you differentiate slow waves VS versus spike potential? Slow waves do not cause calcium ions to enter the smooth muscle cells. They don't have calcium channels, they just have that sodium channels, and that also not to a very vast extent, just a few of them. However, the spike potential causes a lot of calcium ion. So some smooth muscles of the GI tract may exhibit tonic contraction. And what is tonic contraction? It is basically continuous contraction, rhythmic contraction, and this is not with the basic electrical rhythm of the slow waves. It can even last, but it usually lasts several minutes or even hours. And the tonic contraction often increases or decreases in intensity, but it usually continues for a longer time. So the tonic contraction can be sometimes caused by the repetitive spike potential, or to a greater frequency. The greater the degree of contraction, the greater would be the tonic contraction. And certain other things like hormones or your nervous system can also activate them. And the third cause for tonic contraction is, of course, the entry of the calcium ions into the interior of the cell, that can bring about changes in the membrane potentials. So these are the three methods by which the tonic contraction occurs: continuous, repetitive spike potential, hormones or other factors, and then now from pharynx to anus, you have almost similar structure of the GI, that five layers that we just did. Exception is that there is no serosa on esophagus and in the distal. So this is the last, I guess, or the second last slide, just summarizing the what is the digestive tract, and the organs which are involved in breaking down of the food into different molecules that can be. So we have ingestion, from ingestion to the mixing and movement to digestion, absorption, and defecation. Everything is brought about by. So they are, if we just see digestion, we can divide it into two stages. First of it would be the mechanical and then the chemical stage. Mechanical digestion is physical breaking down of food, okay, into smaller particles, either by your teeth, your salivary glands, and your stomach, your churning contractions, okay, and then it serves to expose more food surface to actions of the. And then you have a chemical digestion by which a series of reactions would be occurring, hydrolysis, breakdown of macromolecules into their monomers, okay? Or the hydrolysis carried out by digestive enzymes, for example, salivary enzymes, enzymes coming from the pancreas, from the small intestines, and certain enzymes, certain foods, of course, are absorbed through enzymatic actions, for example, vitamins, minerals. So chemical digestion, you can see carbohydrates. Step one would be degraded into, broken down, sorry, into disaccharides and then monosaccharides, and similarly proteins from peptides into amino acids, and lipids from diglycerides into then monoglycerides. And so the processes in digestion are motility, the muscular contractions, okay, that break up the food, and then secretion. You must know these terms because they would be mentioned again and again. Secretion is the release of enzymes and hormones that would aid in digestion, and then there's membrane transport mechanisms. You all know active transport, what is facilitated transport. We'll be mentioning it when we'll be mentioning certain type of digestive processes taking place. So this is the organization of the alimentary canal that is continuous. It is a tube that begins at the mouth and which includes the mouth, pharynx, esophagus, stomach, small intestine, large intestine. And then you have the accessory organs that aid, and without it, digestion would not be that helpful. That is the teeth, the tongue, the salivary glands, liver, gallbladder, and the pancreas. So this is the end of the slide. This was just an introductory lecture, the start of the module. I hope everything opens physically soon so that we can just do it, the practical aspect as well, and the physical aspect. It will be more easy for us and you to make this possible and to understand things. So see you guys again in another lecture.